Welcome to our educational video on apoptosis. Apoptosis is a pathway of cell death in which cells activate enzymes that degrade the cell's own nuclear DNA and cytoplasmic proteins. This leads to the breaking off of the cell, forming edible fragments consumed by phagocytes. Apoptosis is a form of coordinated and internally programmed cell death, and there is no inflammation unlike necrosis. Morphology of apoptosis. Apoptosis involves single cells or small clusters appearing as shrunken eosinophilic masses within normal organelles. Apoptotic cells undergo chromatin condensation, DNA fragmentation, and shrinkage, forming apoptotic bodies. These bodies are quickly extruded and phagocytosed without inflammation. Apoptotic cells can be identified and counted using various methods, H&E, and chromatin condensation staining, flow cytometry, DNA change detection by in situ or by gel electrophoresis, and annexin 5 as membrane marker. Apoptosis occurs physiologically during embryogenesis and involution of thymus, and pathologically in tumors, immune responses, AIDS, and degenerative CNS diseases. The mechanism of apoptosis. Apoptosis is regulated by biochemical pathways that control the balance of death and survival-inducing signals and ultimately the activation of enzymes called caspases. Caspases were so named because they are cysteine proteases that cleave proteins after aspartic acid residues. There are two pathways, the mitochondrial pathway and the death receptor pathway, which converge on caspase activation. The end result is clearance of apoptotic bodies by phagocytes. First, the mitochondrial or intrinsic pathway. The mitochondrial proteins, including cytochrome C, induce apoptosis when released into the cytosol due to changes in membrane permeability. The BCL2 family proteins regulate this process, with pro-apoptotic proteins promoting permeability and anti-apoptotic proteins protecting cells by maintaining mitochondrial membrane integrity. When cells receive signals to die, like growth factor withdrawal, DNA damage, or protein misfolding, the BH3-only proteins are activated. These BH3 proteins activate pro-apoptotic factors like BAX and BAC. BAX and BAC then dimerize, insert into the mitochondrial membrane, and create pores, leading to cytochrome C release and caspase activation, ultimately resulting in apoptosis. Once cytochrome C is in the cytosol, cytochrome C binds with the PAF1 and procaspase 9 to form the apoptosome, which activates caspase 9. Activated caspase 9 then cleaves and activates executioner caspases, which then sequentially activates all other caspases or activation of a caspase cascade, which lead to nuclear fragmentation and formation of apoptotic bodies. The death receptor or extrinsic pathway of apoptosis is triggered by the binding of specific ligands to death receptors on the cell surface. Death receptors are cell surface proteins belonging to the TNF receptor superfamily, characterized by a death domain in their cytoplasmic tail. Ligands and their activation. There are two ligands. Fas ligand, which are expressed on activated T lymphocytes, which binds to Fas receptors on target cells, leading to receptor clustering or trimerization. The next is TNF ligand, which binds to TNFR1 receptor, initiating similar process. When ligands bind to their death receptors, like Fas ligand on activated T cells binds to Fas receptors on target cells, the death domains recruit adapter proteins such as Fas-associated protein with death domain for Fas, and TNF receptor-associated death domain for TNFR1. These then recruit and activate procaspase 8 and 10 to active caspase 8 and 10. Caspase 8 and 10 then activates executioner caspases like caspase 3, 6, and 7. The activation of the caspase cascade leads to the breakdown of cellular structures, nuclear condensation, DNA fragmentation, and finally the formation of apoptotic bodies, which are then phagocytosed by neighboring cells or macrophages, avoiding inflammation. In either pathway, after caspase 9 or caspase 8, N10 is activated, it cleaves and thereby activates additional caspases that cleave numerous targets and ultimately activate enzymes that degrade the cell's proteins and nucleus. The end result is the characteristic cellular fragmentation of apoptosis. Apoptotic cells or bodies are cleared by phagocytes through beat-me signals. Phosphatidylserine, normally on the inner membrane, flips to the outer leaflet in apoptotic cells, signaling phagocytes. This externalization of phosphatidylserine acts as primary eat-me signal recognized by phagocytic cells like macrophages. Once recognized, apoptotic cells are rapidly engulfed by phagocytes in a process called efferocytosis. The efficiency of this process ensures that apoptotic cells are removed before they can lyse and release potentially harmful contents into the surrounding tissue. This prevents secondary necrosis, which would lead to inflammation. Once inside the phagocyte, the apoptotic cell is degraded within phagocytosomes, where lysosomal enzymes break down the cellular components. If you found this video helpful, please like, share, and subscribe.